Hello, everybody. I'm back. Dr. K.K. Agarwal, President, Heartcare Foundation of India and Confederation of Medical Association of Asia and Oceania. You are watching Med Talks with Dr. K.K. Agarwal, COVID edition 44th episode. Each episode, we are talking about something to take the fear out from the mind of the people. Let's go to the headlines of the day. Don't worry, 70,000 people have been affected in India in the last 24 hours. 65,000 of them have also recovered. WHO cautions against relaxing restrictions too early. We at this moment feel that if you are going to relax restrictions, number will never come back to normal. If your aim is to wait for a vaccine, you need to control your pandemic strictly. Delhi has started its third zero surveillance today. Good news. Second zero surveillance, 28%. First zero surveillance, 23%. Let's see what the third zero surveillance has to say. A study from Telangana has shown a high viral load in asymptomatic patients. High viral load means more seriousness. We don't know. High virus load means more contagiousness. We don't know. High virus load means a live virus or a dead virus. We don't know. In a WHO Global Pulse survey, 90% of countries reported disruptions to essential health services since the COVID-19 pandemic. Let's go to the topic of the day. All about coronaviruses. We want to go to very basics. What this pandemic has done is has taught us a lot of virology, a weak wicket in our MBBS time, because virology, we always thought that this virology is not available in our lab, so we never bothered about to read about virology. But a time has come for us to understand in detail about any virus, potential pandemic causing ability, and vaccine producing abilities against that virus. To discuss, I have with me an expert, Dr. Let me introduce Dr. Pavitra Venkata Gopalan. She is a PhD on coronavirus studies from Arizona State University. I hope she knew during her PhD that such thing is coming. She is an expert in the molecular biology of the virus. She is the director of two labs, Care Health in Chennai and Molecular Solutions in Bangalore. Both of them are NABL accredited molecular biology in Bangalore. So, Pavitra ji, virus is something which most of the doctors today are relearning. This is something people used to say, oh my God, this is just a viral disease, don't worry. We did not know, we did not read about it, because if you treat 1 week, if you don't treat 7 days, that was the dictum. No antibiotic, no treatment, it will be all right by itself. But this virus has taught us different behaviors. This virus has taught us sometimes I can behave like HIV, reduce CD4 cell count. Sometimes I'll behave like a bacteria, increase your procalcitonin. Sometimes I'll behave like a immunological reaction. So I will increase your acute phase reactants, but will not increase platelet count. Sometimes it says I will behave like a thrombogenic activity, increase your D dimer, but will not decrease fibrinogen, but will increase fibrinogen. Sometimes it says I will cause you hypoxia without rise in loss of consciousness. So from hypoxia to happy hypoxia. And sometimes it says I will produce a suicidal cytokine cells to cause cytokine storm and kill everybody. So therefore, this virus basically has come as a mysterious virus, and it has taught us all aspects of virology. And this
Certainly.
virus now is also talking us a lot about its components, which gene to test, which RNA to test. We never bothered about. We used to only listen to positive or negative. Now this virus is telling us high viral overload, low viral overload, CT value in RT-PCR, and this virus is now telling us everything about how to make a vaccine because so many countries are making a vaccine against various components. So I will go back to very simple thing is, first of all, the first 5 minutes I want to spend on yourself. Why do
Sure.
you choose to study coronavirus? And you ever realized that this is going to be the situation in India once?
When I applied for graduate school, 2005 is when, 4 is when I applied. At that point, SARS virus had just come and had been controlled. So it was what was considered a glamorous field to work in, and there was a lot of funding. So it was very interesting for me that I was able to get into a lab that was what is called a hot area of research, and we studied the virus. And while theoretically we knew that this virus had the potential to become a pandemic, especially based right out of SARS, we didn't expect to see it so soon in our lifetimes.
So when we talk about, let me give you an example that when you have a adenovirus of chimpanzee and you put the spike protein of this virus and you say adenovirus 5, you're making a vaccine out of it. And if you can create a vaccine out of it artificially, which is adenovirus, I normally say adenovirus की जैकेट में आपने कोरोना वायरस के बटन लगा दिए.
Yeah,
I mean, you
yes,
are, I mean, just
yes,
general public को समझाने के लिए that what you are doing is you are exchanging the buttons and putting either a recombinant button or a virus button into that.
yes.
Don't you think it's a new virus then? If you have a, don't you think that that becomes a new virus, whether dead or alive? Because I think that adenovirus which you are injecting is live attenuated
Right.
adenovirus with the spike protein of a coronavirus.
That the adenovirus that is used is an attenuated strain. It
Yes,
doesn't replicate quite as efficiently in humans. So that's
agreed.
the safeguard that we have against it becoming a new virus causing more trouble.
Agreed. But then that means experimentally formation of a new virus is possible because you are using a attenuated adenovirus with a spike of a new virus, which now becomes a vaccine virus, but that's a new virus. So therefore, theoretically, people ask, is it possible to make a virus, a hybrid virus in the lab? It's possible. I'm only talking theoretical.
Yeah, it is. It is possible. People have done some studies using a coronavirus protein in a vaccinia virus to study different functions and structures. Ever since recombinant DNA technology was available, theoretically, a lot of these things are possible. Whether they are practically possible, achievable, are is a completely different ballgame.
Okay, let me come back to the understanding of a virus vis-a-vis understanding of a surface antigen virus, that is hepatitis B. We say there is a surface antigen, and we say there is a core antigen, and then we say there is a nuclear antigen there.
Yeah.
Similarly, when we come back to this virus, we say that it has a membrane, envelope, spike, and a nuclear component. Can you
Yes.
describe a little bit about that? What is the role of the nucleic component? What is the role of a membrane?
Yeah,
What
I
is the
will.
role?
So picture the virus to be a ball, like a football, okay? And on the football, you have different proteins sticking out, like spikes. So it'll look like this. And the ones that are sticking out predominantly is called the spike protein. This protein, if you can imagine, it looks like the ball is wearing a crown. So in crown in Latin is corona, and that's how this virus got the name coronavirus. That is the spike protein. And then the viral envelope is made up of one protein, that is where the structure on which the spike protein is stuck on is made of the membrane protein. It's called M protein. And the M protein forms the backbone of the viral structure. Then there is a small protein called the envelope protein, that accidentally gets incorporated during the time of assembly. When you compare the amount of envelope protein in the cell versus the virus has very small amounts of it. So we think it just accidentally comes and forms a part of the viral envelope. And inside this ball is the most important part which allows the virus to replicate, which has the viral RNA. This virus has no DNA. This virus only has RNA, and that is held stably in place by this protein called nucleocapsid. The nucleocapsid protein, the membrane protein, and the spike protein all have different parts that interact with each other to form this viral envelope. So, in a brief overview, this is what the virus is made of.
So therefore, each one will have a specific action purpose.
Yes. Yes.
So therefore, what is the purpose of RNA? What is the purpose of nucleic capsid? What is the purpose of envelope? And what is the purpose of membrane and a spike?
So, like I said, we'll start with spike. We'll come from outside to inside. We'll talk about it as though we are the virus that is entering the cell. So, if you have a cell and you have this viral ball with a spike protein, it comes across the cell, and if it finds a receptor, so this receptor spike protein interaction is like a lock and key mechanism. Not every virus can interact with every receptor. It finds a specific receptor and the spike protein binds to that receptor. It says, okay, great, I'm binding to the receptor. And the spike protein has another region called membrane fusion region. So this viral envelope, the viral backbone of the viral envelope, fuses with a cellular envelope, and it opens up the viral ball and releases the viral genome into the cell. At this point, the membrane protein doesn't have any functional purpose. When the inside of the cell, which is the viral RNA and the nucleocapsid, enters the cell, the nucleocapsid, so the cell is also very clever. It keeps detecting foreign RNA and DNA that comes into the cell. The virus is also very clever, where it protects the viral RNA so that it is not destroyed by the cell. It will stop what is called interferon activity, and the viral RNA will actually resemble what a cellular RNA looks like. And what the cell will think is, oh, here is some RNA, let me make the protein out of it. And the host cell, the infected cell, will start making all the viral proteins from the genome. Once all the viral proteins are made, they start to assemble and more viruses are being made. The membrane protein, nucleocapsid protein, envelope protein all start forming the viral ball and spike protein, and then they are sent out of the cell via the normal transport machinery. So even if a single virus enters the cell, the virus fools the cell into making multiple copies of itself and then releases multiple viral particles outside.
So when we talk about this, so therefore, what is in terms of pathogenesis? Virus has entered, it has replicated. Now this virus has to cause some damage in my body. So I normally say the virus does a Reiki. Reiki means it identifies where are the receptors and it enters. So wherever there are ACE2 receptors, it enters there. Now it has to cause some damage.
Yes.
And damage today we know is it produces cytokines IL-6 and TNF.
Right.
Okay. Which are pathogenic further. So ultimately it will release IL-6 and it will release cytokines. So I'm talking about that out of these components of the virus, which components is responsible for what disease? Is it only spike protein which causes illness?
No.
Is it the RNA? Is it the nucleic? Is it the nucleocapsid which is causing release of cytokine? What is causing the release of cytokine?
Some of the non-structural proteins are thought to cause the release of the cytokines, especially the nucleocapsid protein we know affects the interferon activity of the cell.
So basically, it is the nucleocapsid portion of the virus which will cause gamma interferons.
Yes.
Which will release gamma interferons, and that will cause IL-6 and TNF production.
Oh, sorry. No, I'll have to rephrase it. It doesn't cause the release of interferons. It reduces the interferon activity of the cell.
Okay.
So the cell is still thinking it is not infected. Eventually, when the cell realizes it is infected, it goes and releases a lot of cytokines to fight the viral infection.
Okay, so these cytokines are produced as a not as a inflammatory but as an anti-inflammatory response. That's
Yes.
that is a protective response.
Yes.
So therefore, protection of IL-6 and protection of tissue necrosis factor is for its they are basically for protecting the body at that particular moment.
Yes.
So now again, I mean again why I am asking is in a say for example when we say E antigen, S antigen, and core antigen, they appear at a different time, disappear at a different time in hepatitis B. Here, do we have any data which says which antigen will appear first when we talk about the detection? When we are doing a RT-PCR test, we are doing a gene test.
Right.
So in RT-PCR, we are checking either ORF1A, ORF1B, RDRP, S gene, N gene, and E gene. So E gene is common to all coronaviruses.
Yes.
Okay, now we are talking about the gene which is either we are checking a S gene or we are checking a N gene.
Right.
Is there a timing of them which will appear when, which will disappear when?
The first viral protein that appears that you can detect by real-time PCR is ORF1A, 1B, or otherwise known as RDRP. And then S, M, N, E. That's the order in which it appears. So you're actually looking for
first is RDRP, then
RDRP, then S, M, N, E. This is the order in which it appears.
Yeah, but without E, you are not testing. Once you have checked, if E is absent, then you say don't go to the step 2. That means by the time E has appeared, all have appeared.
All have appeared, yes.
So therefore, that means you will be missing a lots of infections by checking E first.
You're checking. I have not seen a single PCR that doesn't check, a confirmatory PCR that doesn't check RDRP. Earlier, there was a screening PCR that only checked E, but that is no longer being used, as far as I have come across.
So nowadays, there can also be a single gene that is RDRP. You don't test.
They're checking for both because you're looking for both ends of the genome, almost extreme ends. So you want to see what you're seeing is a part of something or if the full genome is getting replicated. So you're looking at almost the beginning and the end.
So you are checking RDRP and E together.
Yes.
For replication with that. So yes. you are saying is RDRP should, for example, some people are checking only S protein along with E. That's not recommended then.
That is not the protocol for real-time PCR. People are checking for the S protein for the other antigen tests, but not for your real-time PCR.
And what are we doing in the rapid antigen? What are we testing there?
Rapid antigen, I am also not very clear about it. I'm very sorry. Because I'm not convinced a single rapid antigen does a good job of screening any of the proteins. What the kits that I have seen are testing for spike protein simply because that is the one that is maximally exposed to the cells.
So therefore, if RDRP is positive and S may be negative, there may be a situation when RDRP is positive and S is negative. Therefore, they will miss lots of cases.
That's why the rapid tests are not reliable.
So that is so when we come back to, I want to come back to a little bit more on a CT value. When we are checking a cycle threshold value, and we are talking about RNA is converted into DNA, and then DNA we amplify the gene. And what is the error of this CT value, which basically tell us the virus load? And we know viral load may not be associated with contagiousness, may not be associated with it may not be associated with severity. But if I'm talking about a reinfection today, when 4 cases have happened, we have in Netherlands, we have in Belgium, we have in UK, this Hong Kong and USA. Do you feel that in a post-COVID phase, that means after 2 months or 3 months, if a patient antigen and antibody are positive, both of them are positive, that means his antibodies are positive, that means he has recovered, and his antigen is also positive and he's symptomatic, and he has 3 CT value available. First CT value was say 30, first CT value was 14, then the second CT value was say 32 before he became negative,
Right.
That means his CT value, once he became negative, he has crossed the threshold. And
Right.
now 3 months later, he again has an infection with a CT value of say 20. Do you say that we can suspect a reinfection in such a situation?
Theoretically, yes. Mainly because antibodies don't last very long after a COVID infection or any coronavirus infection for that matter. So I don't know if the antibodies will last when you test it the second time. You may have a good antibody response 6 weeks after the infection, but there are studies that show antibodies don't really last after about 2.5 months.
Okay, in
So,
that situation, how to check for the cellular immunity? They may not have a humoral antibodies. They may
right.
not show the IgG antibodies, but they may still have because a Singapore study has shown that you have a cellular response even lasting today of SARS-1. So how do we check for a cellular immunity?
I have no idea. I am not an immunologist. I cannot come up with methods for testing cellular immunity.
So what is the cellular response? What is the cellular response of this virus?
So we don't know. We've not really studied so much about the immune response of this virus in humans, simply because we've never caused such a great infection in humans before. 1 in 3 common colds is caused by a coronavirus, but other than that, we didn't really care too much if a person recovered, and that was it. We're still learning what the T cell response for this infection is, what the memory T cell response is, and things like that. The COVID.
Why does it reduce CD4 count?
I don't know. There is a lot we don't know.
There is a lot we don't know because
there is a lot we don't know.
Do you think that being a RNA virus, it will be difficult to make a vaccine?
No, I don't think so. Because while a similar RNA virus, not very similar, but another RNA virus that causes a lot of problems is influenza virus. That people are having trouble having universal vaccine because there is a lot of antigenic shift and drift. But in a coronavirus, the genome is relatively stable because the genome is extremely large. It is the largest of all known RNA viruses. If it mutates so much, it is not going to be able to survive. So it's fairly stable.
What is the percentage of mutation? How many genes should change to call it as a new virus? Because this virus which came in Hong Kong, this says 24 genes have changed, but still it is a COVID. So how many genes should change to become a new virus? Is there a number?
It's not the genes that changes it. It's how closely or distantly related they are to a previously known strain. In terms of structure and in terms of the viral protein structure and function.
And how infectious is this coronavirus with each mutation? That means I'm again coming to the same D614G mutation, which said was more in Europe. It has come to Malaysia. Right. In some cases, maybe there in India. And they said that D641G is not on the receptor binding domain. Therefore, it may be significant, it may not be significant. Then they say it is. So these types of mutations, can a virus reinfect with these mutations? That means the previous virus does not protect you from a newly mutated virus, yet not a new virus. Like what happens in flu.
A very tricky question. It's a very challenging situation. The answer is we don't know. We don't know what the antibody response generated by the body is. We don't know against what part of the spike protein the antibody is being generated. So we still haven't analyzed what the antibody is binding towards. So maybe these single site mutations will have an effect on pathogenicity. Maybe it will not. We have a lot more data than we have understanding and analysis.
So we are getting a lot of information from our guest, Dr. Pavitra, but let me have more discussion after a short break. You're watching Med Talks with Dr. K.K. Agarwal, 44th episode, and 7 p.m. show every day. And I have with me Dr. Pavitra. She is a PhD on coronavirus studies in India, an expert on coronavirus. We have been talking about a lot about what coronavirus is, trying to find out what are the unanswered questions in this particular coronavirus. I'll come back to some more things. Some people are trying to make a vaccine out of the tip of the spike protein. They're further dividing and they say tip of the spike protein may be more antigenic than the whole spike protein. What is this tip business now? We have been confused about the spike and now they say tip of the spike. What is that?
So you have these modeling studies that try to figure out which part is exposed to the cell the most, which part potentially has the most antigenicity. I don't understand the bioinformatics of how you determine whether one portion is more antigenic than or not, other than the fact that it may be more glycosylated in these places. Glycosylation is easily detected by the body than non-glycosylated regions. So there are theoretical studies that predict these kind of domains. So you pick the best domain that fits and then you model it in your vaccine vehicle and develop your vaccine.
So if I want to make a vaccine, so I can use a live vaccine, which is a human challenge. I can use a live attenuated vaccine. I can use a killed vaccine. Then I can use a adenovirus with a spike of this or the spike proteins peak into that, which can be the natural virus or a recombinant virus portion. So I can have a completely recombinant virus without a virus. And what is this VLP, virus-like particle, and what is this DNA domain of Zydus, and what is this virus-like particles which are being used to make a vaccine?
Virus-like particle is very simple. It's basically the virus without the RNA inside it. So it's just the protein that's being given to you. It cannot replicate. It enters your cell because the spike protein is there. And the body also sees the spike protein and then makes antibodies against it.
Okay, so VLP will be a recombinant or it can be a virus made out of RNA has been taken out.
So you develop it recombinant in a recombinant fashion. You don't take the virus, remove the RNA, and then give it.
How do you make a DNA vaccine, which DNA vaccine platform which Zydus is trying to do?
Yeah, so you encode the sequence that codes for the part that is you think will have the most antigenicity, and then you load it in a vehicle and they're like a nanoparticle, and then you inject it as the vaccine. Then your cell will convert that DNA into RNA, and then the RNA will be made into protein.
Okay, so that's how they use it. I'm coming back to, because the time is less, some rapid fire questions. And how does this drug Remdesivir act?
Remdesivir acts against RNA-dependent RNA polymerase. It is a nucleoside analog where it will it will reduce the activity of the RNA-dependent RNA polymerase.
What about Favipiravir?
All of them work in the same fashion.
And out of the two, are they more effective when they use early? Because we say after 9 days, the virus becomes non-replicative. Means what? Because antigen may be positive, RT-PCR may be positive for 3 months or 4 months, but the virus is non-replicative. Means what? What is remaining? Is it a dead virus?
There is a statement that is going around saying remnants of the viral RNA. Even I don't know what that means. Either your virus is replicating or not. So I don't know what this remnants of this virus is.
Then what is positive then? Then what is positive after 9 days? What component of the virus is showing RT-PCR positive?
They check for RDRP and E. But there is no clear understanding or a theory on what is that is turning up positive, especially if a person is completely cured, there is no symptoms and things like that.
No, because what is happening is we are getting post 9th day, we are getting different types of phenomena. We are getting some cases where they have a positive antigen, and this positive antigen is showing recurrent symptoms, but non-contagious.
Right.
And if they are non-contagious, that means the virus is non-replicative.
Right.
But they have something persistent which is showing a report, and that is causing symptoms. So can we explain that?
No, I cannot. I don't know the answer to that. Nobody has given me a convincing answer to that just yet.
Okay. How do we say that this virus is micro versus macro airborne? I'm talking about macro droplet versus micro droplet. So, for example, if I smoke, and if I smoke or if I puff my air, which will be hot, either it will kill the virus or it will convert a macro droplet into a micro droplet. So that means if I speak like ka, it is a macro droplet. If I say dha, it becomes a micro droplet.
Macro droplet.
Pushma sound, or it is a sound which I say mahapran from a alpa pran. So I can convert sounds, and that means it is convertible from a macro droplet into micro droplet. Singing may convert macro into micro droplet. So these micro droplet, how long they can stay in the surface in an open area. I'm not talking about recirculating in a closed, ill-ventilated area.
So this virus is fairly fragile. It is an enveloped virus, which means if the droplet is dried up entirely, it will no longer survive. Now, there are other viruses that will dry up, but when moisture is added, will become active again. This virus is not one of those. There are a lot of studies that say that it will survive in this air for this long, this air for this long. But if you look at the studies, they're all done under experimental conditions, where a specific size of the droplets are being used. When you have these small droplets, and if you live in a hot and humid place and you don't have anybody in contact with it, the droplets exponentially dry much faster. So you cannot put a specific time saying it will dry up within 60 seconds, but your probability of catching an infection from a macro or a micro droplet, if you're not in close contact with it, becomes exponentially less.
So therefore, surface to human transmission is less important than human to human.
Yes.
And this human to human transmission also will be less if you are not in a close contact.
Yes.
And if your contact period is less than 15 minutes, that's what people say.
Yeah. Reducing the probability of you catching the infection through multiple means.
How will a change in terms of mutations? Normally we say when the virus mutates, virus will only mutate if it finds difficult to exist. And for difficult to exist, it has to go for a less virulent, more contagious mutation. So every mutation will be more contagious and less virulent. Am I correct with that?
So I'll have to a little bit, and I can't give you a rapid answer here if you don't mind. So SARS was highly virulent, less contagious, or was mediocrely virulent and not very contagious. And it did not cause too many deaths, relatively. Now, then the next coronavirus that created news was called MERS, Middle Eastern Respiratory Syndrome. It was extremely fatal. 30% of the people infected died. But the virus was easy to contain. So enough mutations have happened in different coronaviruses that this one has found a sweet balance between not particularly lethal and very easy to transmit. So this has found a sweet balance, and that is why it's creating worldwide havoc so quickly. If it was killing everybody, it won't be easy. It won't be difficult to contain it because you'll know when a person is sick and everything. Here, most of the people spreading it are asymptomatic. So it becomes that much more of a challenge to contain this virus.
Okay, so this is a more contagious virus than a lethal virus. And what about a mutation? What makes a virus mutate? And do you think that? So I'll come back to it slightly differently. What is the second wave? Is there a specific definition of a second wave? Is it, say, for example, I take example of Delhi. Okay. In Delhi, we had a wave, and now is it a second wave or is the first wave coming in newer areas? Okay, so if it's coming into, I'm coming to that, or is it a new mutated virus which has entered? Okay, so
Okay.
how do I come to know that a new mutated virus has come as a clinician, as a virologist, as a virologist you would know. As a clinician, how do I suspect that this may be a new strain of virus?
You will have to wait for epidemiological studies from molecular virologists because people are continuing to do surveillance on the strains and their genomic sequence as well. At
So
this
I'm
point, my strong intuition is that this is still the first wave because we also have to take into account that we had a lot of restriction in terms of human movement versus not, and now that is being reopened. So is it an increase in the number of infections because of more exposure to humans, or is it a new strain is the question that we need to answer
last
based on
question. Go ahead, go
sorry
ahead, go ahead. Go ahead.
based on the sequence studies. This we also know that this virus doesn't mutate very rapidly for its own survival. So and also the people that are developing the vaccines are constantly keeping track of the viral genome sequence worldwide, simply because they want to ensure that their vaccine will still be effective a year, 2 years, 3 years from now.
So the last thing which I want to talk about is this virus is causing multiple symptoms.
Yes.
In the same family, if there are 10 people, all 10 of them may have different symptoms as the first
Yes.
symptom onset. Therefore, somewhere or the other, something of the virus may be responsible for a particular type of a symptom. I'm talking, why should it cause somebody diarrhea and somebody it causes headache and somebody it causes this? I have some answers in Ayurveda. I'm not an Ayurvedic doctor, but I
Yeah.
have some understanding of Ayurveda which can explain. But then I cannot explain it in the modern medicine language. Can we explain something? Why
The,
a child? Why a child? Is it thymus in a child? Is it a thymosin alpha in a child who is protecting a child from getting infections? Their infection is only for 2 days. In adults, somebody is getting diarrhea. Somebody is getting eye symptoms, somebody is getting loss of smell, loss of taste, somebody is getting pulmonary, all these
yes.
symptoms may not be present in the same person. And
Right.
in the same family, 10 people may have 10 different symptoms appearing within 48 hours.
The current strongest theory for this is the distribution of the ACE2 receptor in the body. Children are generally thought to have lot fewer ACE2 receptors in their body itself, which actually is the reason that they have very mild symptoms. I am not a big expert on the ACE2 receptor distribution in terms of concentration of the receptors and things like that. So
I
the
know, but
current
if ACE2 was only responsible, then all will have the same degree of ACE2 in their body. So their presentation will be the same, but the presentation is different. I
the
can't say that in somebody ACE2 is more in the brain and somebody ACE2 is more in
absolutely, absolutely.
so there is some mystery still happening as far as this is
We
concerned.
don't know enough about this virus to know why it's causing such variable
I'm going
symptoms.
to come back to the last question is that when we know that in children they have a thymus gland and we know thymus produces both CD4, CD8 cells and the memory T cells and the memory B cells, and that memory disappears by age 12, 13, 14. Do you feel that there is a role of adult vaccination? When we talk about adult vaccination, also adult thymus, as we give them thyroxine, we give them testosterone in the adults. Is there any role of a synthetic thymus vaccine to boost up immunity and to boost up such types of situations as we are facing in COVID-19?
I don't know if it is a risk that we'd be willing to take in term for children, but as you grow older and you get the adult vaccine, the taking additional hormones is not going to affect the immune response of the vaccine that you have taken.
No, what I was saying was that once you have a cellular immunity, when you have a T cell, suppose I'm talking about when you give testosterone in elderly, do you think a time will come people will also talk about synthetic thymus hormones to be given in the elderly like a vaccine?
I don't know. It sounds theoretically. We'll just have to see.
Okay, so that's all for today, and thank you, Madam Pavitra. It was nice interacting with you.
Thanks,
and,
Dr.
Wonderful. I am not an immunologist. I'm not a virologist. I'm a simple cardiologist, but I try to interact with you in the interest of the people, and I hope people who have been watching, and if you are a medical student, if you are a doctor, if you are a healthcare provider, and even if you are a general public who has an interest, kindly watch this video again. It will clarify lots of your doubts. Forward it to your colleague, because we will continue our high-level discussions with the experts in the country. Thank you, Pavitra ji, for your
Thank you,
interactive
Dr.
program.
Agrawal. It was an honor talking to you.
That's all for today. We'll be back tomorrow, 7 p.m. with another show. Namaskar.